Hi. Hi, lovely to see you. So today we are going to do a live stream on the topic of tissue fluid formation. And as in previous live streams, if you've got any immediate questions that you need me to clarify as I'm kind of trying to do the teaching, if you can use capitals, it just helps them stand out for my dear friend who's helping me out with the questions. Do feel free to make topic requests during this live stream and just give me a thumbs up if I've just clarified something for you that maybe you hadn't understood before. I really miss being in the classroom and reading body language and reading facial expressions. That really helps me know whether my teaching is hitting the mark or not. So a little bit of encouragement on that score would be fantastic. So, and finally, we'll have questions at the end on any other topics if you want them. Okay, so let's start with tissue fluid formation. We're going to talk about what it is, how it's made, how the blood's volume is maintained, and what happens to the excess tissue fluid. Now, there are some exam board differences for this topic, and I'm going to run through them. Tissue fluid formation is not in Edexcel A, the Salters-Nuffield specification. It is in all of the others. But it's worth you recognizing that OCR and Edexcel B require you to know about oncotic pressure, which I am going to talk about. I'm going to cover very clearly what is required by all the syllabuses, and then I'm just going to do a quick final PowerPoint slide on oncotic pressure and how it fits in. So what is tissue fluid? Well, basically, it's blood plasma that's been squeezed out of capillaries, and the stuff that plasma that has been squeezed out of the capillaries is bathing every cell in your body. If you ever graze yourself and don't actually burst any blood vessels, you'll notice that a kind of white or clear liquid is kind of sitting on top of the graze. That's tissue fluid. And it's because plasma is that yellowy clear liquid that carries all the other things that is in the blood. So it's worth you kind of recognizing that if you traveled through a blood vessel in a tiny submarine, you would actually be going through yellow liquid, and all of those red blood cells and white blood cells would be flying past you. The reason why we see blood as red when we cut ourselves is because there are so many flipping red blood cells in our blood, but they are being carried by the plasma. And it is fundamentally the plasma that makes up the tissue fluid. We're going to go into that in a little bit more detail. So how is tissue fluid made? This is information that is required by all the exam boards. So here is my very fine diagram of a capillary. We have at this end is the arterial end. It has come from the heart. The blood has come directly from the heart. It is oxygenated, hence red at this end. At this end, it is the venous end because the capillaries at this end will join together to make veins, and the blood will be directed back to the heart and then to the lungs so that it can be oxygenated. So this end comes from the heart, and this end is going back to the heart. Now, hopefully it makes sense. If this end is the arterial end, it is where the heart's contractions really make a difference and really up the blood pressure. It has a high blood pressure. At this end, the blood pressure is very much lower because the blood has had to pass through those tiny capillaries, and the resistance of the blood going past the walls of those capillaries is actually really quite high, which is why the blood pressure decreases so dramatically across the length of a capillary.
Is it the hemoglobin that makes blood red?
Is it the hemoglobin that makes blood red? Yes, it is, because hemoglobin is the pigmented protein, and it's red, that fills the red blood cells, and that is what makes our blood red, because we have so many red blood cells in our blood. If you spin the blood, it's worth you recognizing that the test tube would end up being about half of clear yellow fluid, which is the plasma, and the other half, most of the other half, is the red blood cells. Okay, we've digressed. We're coming back again. Okay, so now we're going to talk about what happens at this end of the capillary, at the arterial end. And the information that you need to know is going to be written on this side of the screen. And what happens at the venous end will be written on this side of the screen.
So the pressure is highest at the arterial.
The pressure is absolutely highest, far higher at the arterial end than it is at the venous end. Now we come to terminology, because you don't call this blood pressure in your A-level exams. You are going to call it hydrostatic pressure. And that is fundamentally saying that the pressure is coming from the fact that the blood is fundamentally made up of water. It's the pressure of the water against the pressure of the water-based blood against the capillary cell walls. And the other thing that I want to point out to you is that this is a pushing pressure. It's a positive pressure, and it is literally positive in terms of any numerical numbers that you will see on an exam board. So at this end, there is a high hydrostatic pressure, and I'll explain why it filters the blood. So it's a hydrostatic pressure. It is actually a positive pressure. So for example, in an exam, it might say, oh, it's plus 5 kilopascals. This is important when it comes to understanding oncotic pressure for you OCR and Edexcel B guys. So this high hydrostatic pressure literally squeezes the plasma out of the capillary wall. The capillary wall does have very fine holes in it. And remember that filtering is separating things by size and by size only. So when we talk about the high hydrostatic pressure filtering the blood, everything is filtered out except for those things that are big: the red blood cells, the white blood cells, and the large soluble proteins. Everything else that is filtered out, equals the tissue fluid. And it's because of this high hydrostatic pressure. Now, at the venous end of the capillary, the hydrostatic pressure is hugely decreased. So the hydrostatic pressure here might be, for example, plus 1 kilopascals, relatively small. But here, importantly, the blood water potential is far less than the tissue fluid water potential, because of all of those soluble proteins that are still in the blood. And therefore, guess what happens? As soon as you hear the word water potential, you can kind of guess what's going to go on. Water moves down the water potential gradient from the tissue fluid into the blood. And this is the arrow that indicates water, as it were, being pulled into the capillary because there is a water potential gradient.
Does glucose get filtered, or is it a big molecule?
Oh, glucose absolutely gets filtered out into the tissue fluid. Glucose is relatively a very small molecule. So in the tissue fluid, there will be things like all the things that the cells need. So oxygen, glucose, amino acids, ions, all of those kinds of things. But the large molecules can't get through. And equally, carbon dioxide would be diffusing through the tissue fluid back into the blood by diffusion because of the concentration gradient. So back to water entering here by osmosis. OCR and Edexcel B, guys, I now want you to hold on to the thought that water is being pulled back in. Here we go. Oh, sorry, final point that I forgot, which is actually really important. Any excess tissue fluid drains into the lymph. There is always a little bit, in healthy people, there's always a little bit of excess tissue fluid. And that we have this weird system called the lymph system, which is an additional set of tubes that go round our body, as well as all of our blood vessels. And it basically acts as a drainage system. And it drains back in to our blood system near the heart, so our blood volume always kind of stays the same. Another question?
So is there a higher water potential in deoxygenated blood than oxygenated blood?
The oxygenation level of the blood will not affect the water potential, because at this point I'm making the assumption that the oxygen is actually attached to the hemoglobin in the red blood cells. So it isn't dissolved in the blood, because water potential a drop in water potential occurs because you have things that are dissolved in the blood. The more that's dissolved, the lower the water potential.
Just to help them remember it, have they got it the right way around?
What's that?
That is there a higher water potential in deoxygenated blood?
Oh, no, I'm not sure what you mean. Hold on. So is there a higher water potential in Oh, no. Oh, what's the difference? Okay, what's the difference? So I'm just going to reiterate, oxygenation doesn't affect water potential. The only thing that affects water potential is how much soluble stuff is dissolved in the liquid. Next question is about, so what is the difference between lymph and tissue fluid? Lymph, because the lymph system is also involved in our immune system, you will actually also find, this is slightly weird, white blood cells in our lymph system. So you would find white blood cells in our blood. You would not find, or hardly any, white blood cells in our tissue fluid, but they would be in our lymph system. Are there any other, I can't think of any other crucial differences between tissue fluid and lymph. I'm kind of scared I'm missing something, but I think I'm all right on that. Okay, we're now going to go on and talk about this information that is required by all exam boards, and hopefully now we're going to connect it seamlessly to oncotic pressure that is required by OCR and Edexcel B. So just to remind you, because of the water potential gradient between the tissue fluid where it's higher and the blood where it is lower, water is being pulled into the blood as a direct result of the soluble proteins that are in the blood. That is oncotic pressure. And notice that oncotic pressure is a pulling pressure, therefore it is a numerically negative pressure. this might be, for example, -3 kilopascals. So here we go. Oncotic pressure. The oncotic pressure is actually there across the length of the capillary. Hence, I am now putting in this other blue triangle here. It is a negative pulling pressure, whereas remember, this hydrostatic pressure was a pushing positive pressure. It is directly as a result, it is because of, this symbol here means because of, it is because of, directly because of the soluble blood proteins. And therefore, because those soluble proteins are soluble, the blood water potential is lower than the tissue fluid water potential. And therefore, water is pulled into the capillary by osmosis. I'm hoping that oncotic pressure now makes sense in the context of the standard information that you need to understand about how tissue fluid is formed at the arterial end and how water or the blood volume is maintained at the venous end. So for you OCR and Edexcel B candidates, at the arterial end, you need to be able to say that the hydrostatic pressure is greater than the oncotic pressure. What numbers did we have in here? We had +5 kilopascals and we had -3 kilopascals. So there is a net pushing out of things out of the blood. The filtering occurs, therefore blood is filtered. And at the venous end, the osmotic pressure is greater than the hydrostatic pressure. So here we have -3 kilopascals, and this was +1 kilopascals. And therefore, overall, there is a pulling in of water. That has the greatest effect. And therefore, water is pulled in by osmosis. I find it really helpful to understand that hydrostatic pressure is a positive pushing pressure and oncotic pressure is a negative pulling pressure. And that helps me get my head around how this all works. Once again, as we've said before, the excess tissue fluid drains into the lymph, exactly as we had in the previous slide. I hope now that this oncotic pressure kind of lays on top of what I had in the previous slide. So, to summarize, at the arterial end, hydrostatic pressure is very high. It's higher than the oncotic pressure. Therefore, the blood is filtered. At the venous end, actually, there is, now if you're not OCR or Edexcel, you would just say, you know, the water potential of the blood is very much lower than the tissue fluid water potential because of the soluble proteins, and therefore water moves back in by osmosis. If you're OCR and Edexcel, you would say that the oncotic pressure is greater than the hydrostatic pressure, and therefore water moves in by osmosis because of the soluble proteins that are in the blood. And I think we're there. Question?
Okay, 2 questions. Is oncotic pressure the same throughout the capillary?
Do you know, it actually is. The oncotic pressure is the same throughout the capillary. Whenever I look up any data, it is factually, it is the same, which is why I've made the arrows the same size.
Okay, second question. So hydrostatic pressure is to do with filtrates and oncotic pressure is to do with water, ?
Yeah, so the hydrostatic pressure is literally pushing the blood plasma through the filtering system that is the capillary wall. It's literally pushing it through. Whereas oncotic pressure is all to do with the movement of water down the water potential gradient by osmosis. I hope that's clarified that. Any other questions? And here I am, I'm now finished. So if there aren't any other questions, we can all go and have a cup of tea. But if there are, start typing. Otherwise?
What occurs with the excess tissue fluid in the lymph vessels?
What occurs with the excess tissue fluid? So the excess tissue fluid, it moves around this lymph system. The lymph system has valves in it, doesn't have any pumping system like the heart. The liquid moves as a result of your muscles contracting and the valves stopping it going backwards. And it's basically channeled up actually to a place very close to the heart where the lymph the lymph fluid drains back into the blood. I hope that's helpful.
Is tissue fluid just water?
Is tissue fluid just water? No, strictly not. It is largely, it's absolutely based on water, obviously. It is the plasma, and therefore anything that is dissolved in that plasma. So dissolved in that plasma would be your glucoses, your other sugars, monosaccharides, your amino acids, your ions, and all of that would be bathing the cells of our body. And all of those things are required by every cell in our body. And equally, carbon dioxide dissolved, not little bubbles, dissolved would be moving from the cells of our body back into the capillaries by diffusion, going from a higher concentration to a lower concentration.
Okay, a few more questions.
Okay.
Do the hydrostatic and oncotic pressure occur at the same time?
Yes. Really, yeah, really quickly. Absolutely, yes, they do. Yes, they're just different conditions at the arterial end and the venous end. And absolutely, those two pressures occur at the same time.
At the venous end, is the tissue fluid moving back into the capillary, or is it just water?
Sorry, let me just have a look. Is the venous end, is the tissue fluid move? Right. It's just water. And AQA make a big deal of you knowing that it is just water. OCR is slightly different. They will allow you to say, and it surprises me, I'm going to be honest, they allow you to say that fluid moves back in. It is just the water because it's a water potential difference. So yes, certainly if you're AQA, I would be very careful about your terminology and saying that water moves back in. I have seen mark schemes absolutely require you to know that, but not so much with the other exam boards, particularly OCR, which I do know their exam questions particularly well, and the same with Edexcel. Eduqas, it's a struggle to look at the questions, so I don't know so much with Eduqas, I'll be honest.
Is osmotic pressure the same as oncotic?
Oh my goodness, fantastic. I'm so pleased somebody asked that. It is absolutely the same. I didn't want to muddy the water. Osmotic, the only difference is you talk about oncotic pressure when you're talking about blood because it's caused by the soluble blood proteins. It's the only difference. If your teachers have taught you what osmotic pressure is, oncotic pressure is osmotic pressure in a blood scenario.
How do you answer 6 marks questions on tissue fluid?
How do you answer 6 mark questions on tissue fluid?
Mm-hmm.
You basically talk about the things that I had let me go back to this slide. You say where things happen. You say you talk about at the arterial end. You say exactly what happens at the arterial end, and it's that it is filtering. Yeah, you say the process, and then you say at the venous end, you say what is happening, and you talk about water moving back in by osmosis. So it's about getting the place right and what is going on right, and talking about the different pressures. Well, we've done that. We've talked about the different hydrostatic pressures, and in terms of OCR and Edexcel, you would be wanting to compare the hydrostatic pressures with the oncotic pressures. I hope that's clear. Basically, if you get all that information in the answer, you'll get the marks. The only other thing I can think of is where I've seen questions where they've said, what causes the filtering? Well, I've seen one mark being because of the heart's contraction, which causes the high hydrostatic pressure. But few questions demand that. I hope that's okay.
Is oncotic pressure the only extra thing that OCR need to know about?
Yes, absolutely. Yeah. Yeah. All the exam, I'm literally giving you this knowledge as per mark schemes. I'm, just understanding the level of detail that you are required to know to get the marks. So I am, honing this right down. So yes, stick to the terminology that I've given you, and I can almost guarantee you'll get practically every mark in the question. That's about as good as I'm going to give you. Is that all right? Are we there on the question? Thank you so much. I really do appreciate the encouragement. So thank you. And time for tea by the sounds of it. So again, let me just remind you, I'm going to put up, if you've given me suggestions as to what you want to do next, I'm going to cut that down to 3. There'll be a poll. Please vote so that I'm actually scratching where it's itching on Tuesday. See you on Tuesday. Just let me stop.